Welcome back to all. Today we are going to discuss another part of immunology, that is immunoglobulin, also known as antibody. Antibodies are antigen binding glycoprotein synthesized by plasma cell in response to antigen stimulus. They are also called immunoglobulin because they belong to a group of protein called globulin and participate in immunosystem, so they are called immunoglobulin. Next point is their most abundant protein component of the blood. They constitute about 20% of the total protein component of the blood plasma. Some antibody present in the blood plasma and some are present on the surface of B cell as B cell receptor. Next, come to the basic structure of antibody molecule. A simple antibody has a Y-shaped structure consisting of two identical antigen binding sites, one at the tip of each arm of Y. Because they contain two antigen binding sites, they are called bivalent. A antibody molecule contains 4 polypeptide chain, 2 identical light chain and 2 identical heavy chain. This light chain contains 220 amino acid and they are around 25 kilodalton in molecular weight. Heavy chain contains 440 amino acid and their molecular weight is 50 kilodalton. They form heterodimer structure. This light chain joined to heavy chain via disulfide linkage and some non-covalent bond. And 2 heavy chain also join each other by a disulfide bond and non-covalent linkage. In mammals, there are 2 kinds of light chain. One is kappa, one is lambda. Each species produce both the kind of light chain, but each immunoglobulin have either kappa light chain or lambda light chain. And there are 5 different types of heavy chain. These 5 different types known as isotypes. According to the different kind of heavy chain, immunoglobulin divided into 5 types. This table shows the immunoglobulin heavy chain isotypes. Immunoglobulin M contain heavy chain mu, D contain heavy chain delta, G contain heavy chain gamma, A contain heavy chain alpha, E contain heavy chain epsilon. Minor difference in the amino acid sequence of the alpha and gamma heavy chain leads to further classification of the heavy chain into subclasses. For example, in human there are 2 subclasses of alpha chain and 4 subclasses of gamma chain. Both light and heavy chain have variable sequence at their amino terminal end but a constant sequence at their C terminal end. Light chain have a constant region about 110 amino acid and variable region of the same size. The variable region of heavy chain is about 110 amino acid long, but heavy chain constant region is 3 to 4 times longer. N terminal end of both light and heavy chain come together to form antigen binding sites. This is the common structure of a simple immunoglobulin. Each immunoglobulin contains 4 polypeptide chain, 2 light chain and 2 heavy chain. These light and heavy chain join together by disulfide bond and many other non-covalent linkage, and 2 heavy chain join together by disulfide bond and many non-covalent linkage. Amino terminal end of both light and heavy chain from antigen binding site. This region is known as hinge region, which provides flexibility to the immunoglobulin molecule. This is the common structure of a immunoglobulin molecule. Actually, the structure of immunoglobulin was elucidated by 3 scientists independently. One scientist Protot digest immunoglobulin by enzyme papain. Through papain digestion, the immunoglobulin molecule digested into 3 segments. This is the papain digestion product: 2 fragment antigen binding site and 1 fragment crystallization. Then another scientist Edelman digested this immunoglobulin by a chemical known as beta-mercaptoethanol. Through this digestion, there are 4 polypeptide chain are produced. These 2 are long polypeptide chain known as heavy chain and 2 are short known as light chain. From this result, they concluded that each immunoglobulin molecule form of 4 polypeptide chain. Next scientist was Nissenhoff. They used pepsin to digest immunoglobulin. Through pepsin digestion, they produce a combined fragment consisting of 2 fragment antigen binding site. And fragment FC, that is fragment crystallization fragment, is not produced through pepsin digestion because it is digested. Now coming to immunoglobulin domain. Actually, in native form, immunoglobulin present in tertiary structure. Each immunoglobulin molecule folded to from many domain. These globular structure are called immunoglobulin domain. This is the hinge region which provide flexibility to the immunoglobulin, and this region is rich in amino acid proline. Now coming to different classes of immunoglobulin which are very important. There are 5 classes of immunoglobulin in mammal. These are IgA, D, E, G and M. Now come to the first class of immunoglobulin, that is IgG. It is the most abundant immunoglobulin in the serum, consisting of 80% of the total Ig. There are 4 subclasses of this IgG. The half-life period of this immunoglobulin is 23 days, but in Ig3 it is 7 days. The most important point of this immunoglobulin classes is it is the only class of immunoglobulin that can cross placenta and provide immunity to the fetus from mother. Next one is IgM. It is the pentavalent immunoglobulin. IgM is the first class of antibody produced by a developing B cell. It is consisting of 5 to 10% of total Ig. It is form B cell receptor on the B cell surface together with IgD. Its molecular weight is 180 kilodalton. It is in plasma. It is present in pentameric form. There are 5 monomers joined by a J chain. This is the first class of immunoglobulin produced in primary response, and in units it is the first antibody is produced. It involved in complement fixation. Next one is IgA. It constitutes around 10 to 15% of total Ig in serum. It is the predominant, most important point, it is the predominant Ig class in the external secretion, that is the breast milk, saliva, tear contain this antibody. There are 2 subclasses of this IgA. One is IgA1, IgA2. It is the most predominant and 93% of IgA1 found in blood serum. Its half-life is 5.5 days. It is the most predominant antibody in the external secretion. It contains 2 additional polypeptide. One is secretory component, one is J chain. There are 2 monomer joined by a J chain to form dimer. Next one is IgE. It involved in immediate hypersensitivity reaction, which form allergic reaction. The symptoms are high fever, asthma, hives, anaphylactic shock. IgE actually bind to the Fc receptor on the membrane of blood basophil and tissue mast place. When they are cross-linking with antigen, that causes degranulation of that basophil and mast cell, produce various vasoactive substances like heparin, serotonin, histamine that produce allergic reaction. Next class is IgD. It is the last class. It constitutes about 0.2% of total Ig in the serum. Together with IgM, it from B cell receptor, and there is no major biological effector function has been identified for IgD. This table show all about different classes of antibody and their characteristics, their molecular weight, their percentage in blood serum, which antibody can cross placenta, which antibodies involved in complement fixation, half-life period of all the antibodies, their percentage of carbohydrate. This table shows characteristics of all the antibodies. To binding antigen, antibody participate in a broad range of effector function that will result in removal of antigen and death of pathogens. The first one is opsonization. Opsonization is a process by which microorganisms and other foreign particles are coated with antibody and other complement and thus prepare for recognition and ingestion by phagocytosis cell. Actually, opsonizing antibody is IgG. IgG bind to the Fc receptor which is present on the phagocytotic cell like macrophages, neutrophil. This binding provide phagocyte a method to capture antigen. This is all about opsonization. Next one is toxin and viral neutralization. Many gram-positive, gram-negative bacteria produce many toxin. This produce pathogenic effect on our body. Immunity against such toxin depend on the production of specific antibody that inactivate this toxin. This process is called toxin neutralization. There are various antibody like IgG, M and A that bind with viruses during their extracellular phases and inactivate them. This is called viral neutralization. Another function of immunoglobulin is activation of complement. Actually, complement are certain plasma protein that can be activated by either binding to certain pathogen or by binding to the antibody. Complement activation, often described as a series of cascade enzymatic event leading to generation of specific complement component that causes opsonization or phagocytosis of the infectious agents as well as directly lyses the organism. IgM, IgG can activate complement system. There are 3 subclasses of IgG. 1, 2, 3 can activate complement. Next function of antibody is immunocomplex formation. The interaction of antigen with antibody may result in varieties of consequences like precipitation and agglutination. Precipitation results from interaction of a soluble antibody with a soluble antigen to form an insoluble complex. Antibody that those aggregate soluble antigen is called precipitin. The reaction of antibody with a multivalent antigen that is particulate result in the cross-linking of various antigen particles by the antibody. This cross-linking eventually result in the clumping or agglutination of the antigen particle by the antibody. Next one is very important, that is antibody dependent cell mediated cytotoxicity. Actually cell mediated immunity is provided by T cell, but this is the way how antibody participate in cell mediated cytotoxicity. Actually, the function of natural killer cell is to destroy malignant cell and cell infected with microorganism. They recognize their target with the help of binding with antibody. In this case, antibody first coated on the infected or malignant cell. Thus, antibody bridges the two cells. This process is called antibody dependent cell mediated cytotoxicity and can result in death of the target cell. This is all about action of antibody. So, the 5 important points of antibody are: IgG classes of antibody can cross placenta. IgA is the secretory antibody present in external secretion. IgE antibody participate in allergic reaction, that is hypersensitivity type 1. IgD together with IgM